have a confession to make. Whenever I see a headline about the gene for X, the gene for intelligence or for liking cilantro, I tend to just scroll right past it.
You've developed a healthy skepticism.
It's not just skepticism, it's fatigue, especially with the brain. It feels like for the last 20 years, we've been promised this clear instruction manual, but every time we open the book, the pages are in a completely different language.
That's actually a very fair assessment of where genetics has been, particularly with complex conditions like autism.
Right. I mean, you take Autism Spectrum Disorder, you look at the research, and it's not one gene, it's not 5 genes, it's hundreds.
Over 100 high-confidence genes identified in just the last couple of decades, yeah.
And that's where my brain just breaks. You have gene A, which does something with, I don't know, electricity in the cell, and gene B, which builds the scaffolding. They're doing completely different jobs.
They are biologically distinct, absolutely.
So how on earth do hundreds of completely different broken parts all lead to the same output? To the overlapping features we recognize as autism? It feels like we aren't looking for a needle in a haystack anymore. We're looking at a haystack that's made of needles.
It is this central paradox of the field. We call it the complexity crisis. We have this long list of parts, but no idea how they fit together to make the machine run differently, or at least we didn't.
Okay, that is a setup if I've ever heard one. You sent me this new paper published in Nature Heavy Hitters involved here. Gordon, Yoon, Bix, and a huge team, and they seem to think they found a way to make sense of the mess.
They haven't just found a way to make sense of it, they've potentially mapped the route. The study's titled Developmental Convergence and Divergence in Human Stem Cell Models of Autism.
Which is a mouthful, but the scale of this thing is what stopped me in my tracks. Usually when we cover these studies, it's like, we looked at a mouse, or we looked at cells from 3 people. This was bigger.
Much bigger. This is one of the largest studies of its kind ever attempted. And they didn't use mice. They used human cortical organoids.
Okay, we need to pause on that term. Cortical organoids. I know the shorthand is mini brains, but I feel like that just glazes over how wild the science actually is. Are we talking about like brains in jars? Are we in a sci-fi movie here?
It's closer to sci-fi than you might be comfortable with.
Yeah.
But no, these aren't conscious brains in jars. Think of them more like 3D clusters of brain tissue.
But where do you get them? You can't just ask 55 people to donate a chunk of their frontal lobe.
That would be unethical, to say the least. No, this is the magic of what we call induced pluripotent stem cells, or iPS cells. The researchers took skin or blood samples from 55 different people.
Okay.
And they chemically reprogrammed those cells to go back in time, essentially turning them back into stem cells.
So you take a skin cell, wipe its memory, and then tell it, you can be anything now.
And then you give it a new set of instructions. Become brain tissue, specifically the cerebral cortex. So you end up with these growing, three-dimensional balls of human brain tissue that carry the exact genetic code of the donor.
That is just mind-blowing. So they have this library of 55 different brains growing in a lab, and these weren't just random people, right? They were picking specific fights.
They were. They selected donors to cover the entire landscape of autism genetics. They had people with those famous heavy hitter mutations, things like 16p11.2 deletion, Timothy syndrome, Phelan-McDermid syndrome.
But these are the rare specific genetic breaks that we know cause autism.
Exactly. But they also included people with idiopathic ASD.
Which is the medical term for, we have no idea why you have this.
Pretty much.
Which honestly is most people with autism.
Correct. So they had the rare genetic cases,
Yeah.
the mysterious common cases, and of course neurotypical controls. And then, and this is the part that really sets the study apart, they didn't just take a snapshot.
They made a movie.
In a sense, they watched these organoids grow over 100 days. They took samples at day 25, day 50, day 75, and day 100.
I want to get into why that timeline matters, because to me, 100 days sounds like nothing. In a human life, that's just 3 months. But in an organoid, what are we actually watching?
You're watching the construction of the cortex. I mean, at day 25, it's a construction site. You have these cycling progenitor cells just dividing rapidly, laying the foundation. It's messy.
Okay.
But by day 100, you have deep layer neurons, you have structures,
Yeah.
you have the beginnings of a real wiring diagram.
So they're watching to see if the building goes up according to code, and this is where they found the pattern that explains the title of the paper, Divergence to Convergence.
Yes, this is the absolute core finding.
So walk me through it. Let's start at day 25, the early days. We have all these different genetic mutations, the Timothy syndrome group, the deletion group. What do they look like?
They look completely different from each other. They're divergent. The organoids with the 16p11.2 deletion are showing one pattern of gene expression. The ones with Timothy syndrome, a totally different pattern. It's chaos.
So at the start, the specific mutation really matters. If you broke the foundation, the house looks different than if you broke the roof.
Precisely. The genetic starting point is driving the bus.
Mm.
But as they kept watching, as they got to day 50 and then day 75 and finally day 100, the differences started to fade.
The funnel?
The gene expression patterns started to look more and more alike across all the different groups. By day 100, it didn't matter as much which mutation you started with. The organoids were all arriving at the same state.
That is weirdly comforting and also kind of terrifying. It's like no matter which road you take, you end up at the same traffic jam.
Biologists have a term for this. It's called canalization.
Mm-hmm.
Imagine rain falling on a mountain peak. The drops land in different spots. That's the divergence.
Mm.
But gravity and the shape of the mountain force all those drops into the same valleys.
And eventually into the same river.
Exactly. The developing brain has a shape it wants to form, and it tries to buffer these genetic errors, forcing them down a common pathway.
But in this case, the river they end up in isn't a good one. What did they actually converge on? What does the traffic jam look like?
They called it diminished neuronal maturation.
Which means they just didn't grow up properly.
They were delayed. By day 100, the control organoids, the healthy ones, were building complex synapses, setting up their ion channels, getting ready to fire signals. The ASD organoids were just lagging. They had less synaptic machinery. The wiring wasn't being installed on schedule.
So despite starting with completely different genetic errors, the net result was we are late building the connections.
That is the convergence.
Okay, but I have to stop you here.
Yeah.
Because there was a huge exception to this rule, and honestly, it's the part that confused me the most.
The idiopathic group.
Right. The people with autism who don't have a specific known genetic mutation, which, again, is the majority of the autistic population. Did they show this same convergence? Did they end up in the traffic jam?
No, they didn't.
See, that feels like a record scratch moment. You have this huge paper claiming to have found the shared path of autism, but the group representing the most common form of autism doesn't fit. Does that mean the study failed?
Not failed, but it reveals a fundamental difference in the biology. We have to distinguish between, say, a sledgehammer and 1,000 paper cuts.
Okay, I'm listening.
The groups that converged, Timothy syndrome, the deletions, those are caused by rare high-impact mutations. That is a sledgehammer to the genome. You break that one gene, and the effect is massive and obvious.
Okay.
Idiopathic autism, what we call common autism, is likely polygenic. It's not one gene breaking. It's thousands of tiny common genetic variances that, on their own, do almost nothing. But if you inherit enough of them, they stack up.
1,000 paper cuts.
And 1,000 paper cuts are much harder to see in a lab model than a sledgehammer wound. The signal is just quieter. It gets lost in the noise of the experiment. So the idiopathic organoids might have been converging, but the signal was probably too weak for this technology to pick up.
So it's not that there isn't a mechanism, it's just that the mechanism is way more subtle.
Exactly. It tempers the headline a bit. We have found a convergence for high-impact genetic forms of autism. We are still squinting to see the map for the rest.
Fair enough. Let's go back to the sledgehammers then. Because the researchers didn't just stop at, hey, look, they're all late. They wanted to know who was making them late. They went hunting for a culprit.
And they found one. They identified a specific gene network, a module, that seemed to be controlling this whole process. They called it Module M5.
M5? Sounds like a secret intelligence agency.
It sort of acts like one. M5 wasn't just another group of genes. It was a network that was consistently suppressed, turned down, very early in the process. We're talking day 25.
So before the traffic jam even happens, M5 goes dark.
Yes. And when they looked at what genes are actually in Module M5, everything just clicked. It wasn't full of genes for building neurons. It was full of genes for managing the DNA.
Okay, explain that.
Think of your DNA as a giant library. It has all the instructions for building a human. But you can't build anything if the books stay on the shelves. You need a librarian to go get the book, open it up, and hand it to the builder.
Okay, that makes sense.
The genes in M5 are the librarians. Technically, they're called chromatin remodelers and transcription factors. Their job is to physically unpack the DNA so it can be read.
So M5 isn't the blueprint itself. M5 is the person reading the blueprint.
Correct. Specifically, they found genes like SRBOB1 and SMARCA4, which are part of a complex called SNF. These are heavy-duty regulators. So if M5 is suppressed at day 25, if librarians go on strike, Then later on at day 100, when the brain needs the blueprints for how to build a
The
synapse,
library is closed.
and the construction stops, or at
Ugh.
least it slows way, way down.
That is a wildly elegant explanation. The early chaos knocks out the managers, the M5 module, and because the managers are gone, the workers, the synapse genes, never get their instructions later on.
It connects the timeline perfectly.
Mhm.
Early gene regulation failure leads to late structural failure.
But, and I feel like I'm playing the role of the annoying reviewer today, how do we know this isn't just a coincidence? Just because M5 is down and the neurons are late doesn't mean M5 caused the delay. Maybe M5 is just another victim of the chaos.
A classic correlation versus causation problem.
Mm.
And the researchers anticipated you'd ask that. So they decided to sabotage a healthy brain to prove their point.
They framed M5.
In a way, yes. They took healthy neural progenitor cells with no autism mutations, and they used a tool called CRISPR.
CRISPR interference.
Right. Instead of cutting DNA, CRISPR just sort of sits on a gene and blocks it, turns the volume down. So they took these healthy cells and deliberately turned down the volume on the key genes in M5, like one called RBBP5.
They forced the librarians to go on strike in a healthy brain.
And the result was immediate and undeniable. When they suppressed that M5 network, the healthy cells started acting exactly like the autism cells.
The traffic jam appeared out of nowhere.
The traffic jam appeared. Over 1,000 downstream genes were altered. The synaptic genes were downregulated. The ion channel genes were downregulated. They essentially created the molecular signature of autism just by tweaking this one control knob.
Wow. That is the smoking gun.
It is. It proves M5 isn't just a bystander. It is a driver. It's the bottleneck that these different genetic mutations are all funneling through.
So, zooming out, we started with this complexity crisis, the idea that autism is just too messy to understand.
And we've ended with a model of convergence. We now know that for many of these genetic forms, the messiness funnels down into a specific failure of chromatin remodeling the librarians, which then leads to a specific failure of neuronal maturation.
It gives us a target.
Exactly. If we know the traffic jam is happening at M5, maybe we don't need 100 different drugs for 100 different mutations. Maybe we just need to find a way to support that M5 network to, wake the librarians back up.
Which sounds incredibly hopeful. But I can't stop thinking about the timeline we talked about. And this is the part that's, I guess, haunting me a little bit.
The window.
Yeah. We're talking about organoids, day 25 to day 100. In a real human pregnancy, that corresponds to the first and second trimester.
Yes. The critical events described in this paper, the suppression of M5, the start of the traffic jam, they're all happening in the womb.
So if a child is diagnosed with autism at age 2 or 3, biologically speaking, the event happened years ago. The library closed before they were even born.
That is the heavy implication. If the convergence happens that early, does it lock the brain into a certain trajectory?
Right. Is it too late to wake up the librarian?
The pessimistic view is that the developmental window has closed. The structure is built, they can't easily rewire it. But the optimistic view, and the one many in the field are holding on to, is that the brain is plastic. Even if the initial wiring was delayed, the neurons are still there, the genes are still there.
The books are still on the shelf.
They are. If we can find a therapeutic way to reactivate that chromatin network, or to bypass the blockage and stimulate synapse formation directly, we don't know yet. But for the first time, we aren't just guessing at the mechanism. We know where the lock is.
And that's better than staring at a haystack.
Much better.
We'll leave you with that thought to chew on. Thanks for diving deep with us.
Always a pleasure.